So we've talked so far about 3 out of the 4 classes of organic biological molecules. So just remind me, what are the 3 broad categories we've talked about so far?
Carbohydrates.
CC? Lipids, proteins, and carbohydrates. So the last group to talk about in biochemistry for us are the nucleic acids. So nucleic acids are a large molecule. They are made of carbon, hydrogen, oxygen, nitrogen, and also a large amount of phosphorus as well. So that's one of the things that sort of can clue you in as to the identity of a molecule. If it has a lot of phosphorus, it's likely a nucleic acid. And nucleic acids they are our genetic code. Our genetic code is made of these molecules of DNA. We also use RNA to express our genes. So when we talk about the genetic code, we're talking about nucleic acids. All living organisms on Earth have the same genetic code. It's one of the unifying principles of living things on Earth. They all have the same genetic code, made of the same types of molecules with the same components. What our genes really do is control protein synthesis. What does synthesis mean? To create proteins. When we say we have a gene, let's say for eye color, what that really means is that somewhere in our DNA, there's a gene, a sequence of bases in a nucleic acid that tells our body how to make a certain protein that's a pigment that gives our body its eye color. Okay? And there are genes that can turn that on and off, but that's what a gene really is, the instructions about how to build a single protein. And there's 2 types of nucleic acids that are important in living organisms, DNA and RNA. They're very similar, but there are some differences. Does anyone know what DNA stands for?
Oh.
Guys of the DI.
Wait,
No, okay, don't say
Begins
that.
with
the
D.
ribonucleic acid.
Close. Deoxyribonucleic acid. And the difference is in the sugar they have. So they all have sugar as a part of them. And in DNA, it's deoxyribose. In RNA, it's just regular ribose. So that's why they get these 2 different names, DNA and RNA. There's other differences as well. So DNA is found in the nucleus of cells in eukaryotes. In prokaryotes, they don't have a nucleus, so the DNA is found just spread throughout the cytoplasm. Also, there's some DNA in mitochondria. There's some DNA in the chloroplast as well. And if you remember when we talked about evolution, that's because chloroplast and mitochondria evolved from independent prokaryotes that form that symbiotic relationship. So DNA is in the nucleus. It's found on the chromosomes. So chromosomes are made of DNA wrapped around proteins. And DNA is a molecule that stores huge amounts of information in a very, very small space. The elements that DNA is made of are carbon, hydrogen, oxygen, and like I said, nitrogen and phosphorus. It's a very, very large polymer, the largest of the polymers. The monomer, the building block of nucleic acid, is called a nucleotide. So we'll talk a little bit about those building blocks, those monomers. So a nucleotide is the monomer. This is one single nucleotide. Sometimes we draw it using these symbols. And a nucleotide has basically 3 parts. Kind of like an amino acid had 4 parts. Right? The amino group, the carboxyl group, the hydrogen, and then the R group. Nucleic acids, the monomer is a nucleotide, which has 3 parts. So the 3 parts are the phosphate group, this PO4. It's often drawn as a circle. So down here in this model, it's a circle. There's a sugar, which in DNA is deoxyribose. It's a 5-carbon sugar. That's why it's usually drawn as a pentagon. And then there's attached to it what we call a nitrogenous base. And this nitrogenous base is what varies from one nucleotide to another. In DNA, there's four different nitrogenous bases that might be attached here. And we refer to them often by letter or by their names. So the four possible nitrogenous bases are adenine, thymine, guanine, and cytosine. We abbreviate them A's, T's, G's, and C's. Do you remember in that lizard lab when you took that big long DNA sequence and copied it and then pasted it into that other website to help you make the phylogenetic tree? What were the letters that were in that long sequence that you had to copy? Yeah, they were these four bases. What you were looking at in that, you're taking a sequence of these nitrogenous bases from those lizards, A's, T's, G's, and C's, and using them to compare how they're related. The order of, you have about 3 billion of these letters make your genetic code. And you are different from any other person because some of those letters are different in you than they are with somebody else. And those bases are what we call our genes, the order of those letters. It's kind of like an alphabet that you can use to make many different words. It's only this alphabet is just 4 bases instead of 26 in English alphabet. The shape of a DNA molecule is, what's it called?
Oh, it's a corkscrew.
Helio.
Yeah,
You get it
Helix, double helix.
It's called the double helix. A helix just means a spiral. It's called a double helix because it has 2 intertwined spirals. So it forms like, you might think of it as a spiral staircase, or like if you took a ladder and twisted it, that's the shape of a DNA molecule. So the sides of that twisted ladder are the sugar and the phosphate. Those are kind of the backbone. And then the steps of the ladder, those are made of the bases. And the bases on each strand align with each other in a certain pattern. So a DNA molecule has 2 strands to it, and we say that they are complementary. They match up in certain ways. And the way they match up is that if you have adenine on one side of the DNA molecule, you always have thymine on the other. Those 2 bases are complementary. A and T always go together. And G and C always go together. So wherever you have an A on one side, the other side is a T. Wherever you have a C, you have a G, and so forth. The structure of the DNA molecule was sort of a mystery for a long time. Scientists had learned that our genetic information is stored in DNA in the nucleus, but for a while they did not know how all the parts were connected. They didn't know the structure, they didn't know the shape. And scientists were trying to figure it out for a long time. Some scientists proposed a single helix. Some scientists proposed a triple helix, or other shapes. They didn't know if the bases were in the middle or if the bases were facing outward. It was James Watson and Francis Crick that eventually discovered that in 1953. They discovered that the DNA molecule is a double helix, spiral staircase, and they won the Nobel Prize for their work. They're very famous. However, an interesting story, if you are interested in it, there was also a woman named Rosalind Franklin. She was also a researcher. And Rosalind Franklin, her specialty was X-ray crystallography. She would take pictures, X-ray pictures, of crystallized compounds, and you can use that to kind of figure out the shape of something. And so there's a lot of controversy with Watson and Crick and Rosalind Franklin. There was some infighting in these different groups. And the way it sort of happened was Watson and Crick saw some of the X-ray crystallography images that Rosalind Franklin had produced, but they never, she never received credit for helping in the discovery of DNA. She was not awarded the Nobel Prize for that. And there's some bitterness about that. At the time, in the 1950s, there weren't as many women in scientific research. They often were not very well respected by other scientists, and so it's an interesting story about women's contribution to an important discovery and how she helped in her X-ray crystallography for them to find that. So a DNA molecule, here's a model with the sides of the ladder, the sugar and phosphate. The rungs are the bases, A's and T's, C's and G's, and they're complementary. So anytime that a cell needs to reproduce, it needs to completely copy its DNA. And it needs to do it like perfectly, so that there's no mutations. And the structure of DNA allows that to happen. When DNA is going to copy itself, so if you need a copy of this little segment, basically the DNA unzips right down the middle. Okay? The bases are held together with hydrogen bonds, which, as you know from studying water, are relatively weak bonds that can be separated. So the DNA molecule separates down the middle, so each strand is separate. And then new nucleotides kind of fill in the gaps, but they fill in a complementary way. Because you have an A here, only a T matches up with it. Because you have a C here, a G fits in. And after this process completes and you're left here, how do these two DNA sections compare to each other? They're identical. So the DNA has copied itself because it has these complementary bases. This is how a DNA molecule replicates. The bases in DNA come in two groups. So thymine and cytosine are called pyrimidines. They have a single ring structure. And adenine and guanine are purines. And they match up, a pyrimidine with a purine, and they form these hydrogen bonds between each other that hold the DNA molecule together, but can also be undone. Okay? And so you see that shape of this double helix forms the steps of that ladder. That's the genetic code. So like we said, in your DNA, your genes are basically a series of G's, A's, T's, and C's that tell your cells how to make a certain protein and when to make that protein, when not to make it. That's what our genetic code really is. But if you remember back to 7th grade talking about cells, chromosomes are found where? In the nucleus. DNA is a very, very large molecule. It is stuck inside the nucleus. It cannot escape. But it has the recipe to build a protein. But where are proteins actually made and put together? Does anyone remember the name of the cell structure? Brian? Yeah, it's made in the ribosomes. Where are the ribosomes? Sophia? Out in the cytoplasm. So somehow, a cell needs to get the recipe for how to build the protein from the nucleus, where it's stuck in the DNA, out to the ribosome, which is going to actually put the protein together. And the way it does that is using a different type of nucleic acid called RNA. So RNA is another polymer. It's not as large as DNA. It's only single-stranded. It also has a monomer, which is a nucleotide. And it has the same basic parts. You have a phosphate, you have a sugar. It's a different sugar, it's ribose. And then you have a base. Again, there's 4 possible bases that you can have in RNA. 3 of them are the same, adenine, guanine, cytosine. But there's a difference. RNA doesn't have thymine, the T. So if you're looking at the sequence of bases in RNA, you'll never see a T. What you'll see instead is a U, because RNA has uracil as that other base. So here you have a model of an RNA molecule. Again, it has just a single strand of sugars and phosphates, and then it has the bases on the inside. So RNA is a small molecule. It can get out of the nucleus. So when the recipe to build a protein has to be used, inside of the nucleus, the DNA unwinds to show that recipe. And then a molecule called messenger RNA is created. We abbreviate it mRNA. And basically, after the messenger RNA is created, it has a copy of the recipe from the DNA. And that messenger RNA can leave the nucleus. It goes out of the nucleus. Where do you think it needs to go? To the ribosome. It's got to bring the recipe to the thing that's going to actually put the protein together. So it goes out to the ribosome. The ribosome is also made of RNA. So it has the recipe. The ribosome is going to help put it all together. The last thing it needs are the ingredients for the protein. And what do you think the ingredients of a protein are? What are proteins made out of? Amino acids. How many different amino acids are there? 20. And so the RNAs, the messenger RNA has the recipe. It says, okay, I need an argine. I need a glutamic acid. Then I need a leucine. Then I need another leucine. It has all the instructions. And it's transfer RNA molecules, another type of mRNA that actually brings each amino acid in order. The ribosome links these amino acids one to the next, creating this sort of beaded necklace of all of these different amino acids. So these three types of RNA all work together to take a recipe to build a protein from the nucleus and sort of build the protein at the ribosome. Let me finish up with this slide. So we call that process protein synthesis. Our genetic code is determined by the order of those bases, A's, T's, C's, and G's. Humans have about 3 billion letters in our genetic code. And the DNA and RNA work together to actually build the proteins when they're needed. So the basic steps, the DNA gene gets copied to messenger RNA in the nucleus. What's going to be the second step? So we copy the recipe to mRNA, it's in the nucleus. What's it have to do though? It's got to go to the ribosome. So the messenger RNA leaves the nucleus, goes out and attaches to the ribosome. So now we got the recipe, we got the ribosome. What do we need to actually make the protein? We need amino acids. What's going to bring the proper amino acids? Transfer RNA. Transfer RNA brings the proper amino acids. The ribosome starts linking them up until the protein is eventually finished. And when it's finished, the amino acid sequence is released, and it folds up into some crazy shape, and the protein has been produced. And that's how we go from the recipe to build a protein in the DNA to actually a fully formed protein out at the ribosome. Through this series of steps, utilizing DNA and RNA together.